So great thanks to the Academy also to confer this fellowship in 2023. So today I would be talking more about my research, which I've been doing at BRIC-National, which is under one of the plant sciences institution under Biotechnology Research and Innovation Council. And my group has been working in this very crop, Indian oilseed mustard, Brassica juncea, which we know. This crop is quite important for Indian subcontinent for many reasons. I am going to talk more about it. And primarily we work more to understand how the specialized metabolite which are present in Brassicaceae family members are being synthesized and can be used for human values. This crop, I'll just a brief introduction about this crop. This is a very important oilseed crop after soybean. It is cultivated in almost about 1/3 of the Indian subcontinent region. Is mostly in the northern part of our country, and production wise, although it lags behind kind of soybean, but believe me, the oil quantity is almost much higher than soybean, what we get as soybean. And this is a very important oilseed crop in terms of edible oil, and also the oil cake which is left after oil extraction is a very good important for livestock and poultry feed. Unfortunately, our country right now is importing almost about 2/3 of our edible oil through imports, which is a huge burden in our economy. And to meet this task, Indian oilseed mustard, which is almost about 40% of oil is there, it needs to contribute about 20 million metric ton of oilseed production by 2050 or so. Now, there are major areas of improvement which need to be done to achieve this goal. We need to increase the productivity. Productivity basically production per acreage. We can go either through pure line, but it has some limitation that one cannot achieve to a certain level after certain levels. Heterosis breeding, that is basically mostly based on hybrid seed production, is another way to achieve this. And there are some good examples in Indian oilseed mustard, using cytoplasmic miliary system as well as using GMS based or genetic miliary system. Yes, this crop is also infected by or infested by a lot of pathogen and pest. Sclerotinia, in the last 1 decade or so, Sclerotinia stem rot is one of the big problem for this crop, and also Aurobenchia, a root parasite, is coming in a very big way to kill this crop and to damage this crop under field cultivation. My group has been working mostly on improving the quality, oil quality and oil cake quality of this crop, mostly on quality improvement, and I'll be talking about glucosinolate as one of the traits. What are these glucosinolates? So these are the specialized metabolites which are present only in the Brassicaceae family member. All crops like Brassica juncea oilseed, which is a oilseed crop, not only this, mustard family crops like cabbage, cauliflower, all these crops contain these secondary metabolites, specialized metabolites, and these are synthesized from amino acids. So precursor, based on the precursor amino acid, they are named as aliphatic glucosinolates, especially if they are derived from methionine, leucine or isoleucine, or they can be categorized as aromatic glucosinolates, like which if they are derived from methionine and tyrosine. So this is a basic structure of glucosinolate. If you can see here, there is a glucose moiety linked to a sulfonated unit, and this R chain is the one which is contributed by the amino acid. Now, in a single crop, you can have almost about 30 different kind of glucosinolates ranging from these aliphatic or aromatic amino acids. These glucosinolates per se are not so active, but as soon as there is any pathogen attack or any wounding or any crushing of these crops, you found that these glucosinolates are degraded by myrocinases, which are a class of beta-thioglucosidase enzymes, which cleaves these glucosinolates into isothiocyanates form. And these isothiocyanates are the one which are very important for plant pathogen interaction, because they are the one which are toxic to pest and pathogen. So plant have retained them for their usefulness in terms of pathogen or on terms of plant defense. These glucosinolates and their hydrolysis products, especially these isothiocyanates, have various properties, like you can have positive effects, especially some of the glucosinolates which are present in broccoli. Glucoraphanin, they are known to be anti-carcinogenic. Also, they have a lot of chemoprotective properties in terms of when we consume these Brassicaceae family member crops. On the negative side, some of the glucosinolates, especially which is present in oilseed crop like Brassica juncea, they are goitrogenic. You have large amount of progroitin which competes with iodine, and thereby the oil cake which is fed to animals, they make retardation in the animals. So they are goitrogenic. Also the pungency factor, they also reduce the taste of the oil cake to the animal, and as a result, the animal do not like this protein cake or oil cake. So when we screened a very good all Indian oilseed mustard cultivar, and we found that there is very high amount of these seed glucosinolates. As a result, the high amount of these glucosinolates, the oil cake is not very useful for these poultry and livestock, and they are goitrogenic to livestock also. Also, they have very biting taste. As a result, the livestock do not like eating these protein-rich oil cake. Globally, if you see, there is a desire to have canola quality oil which are low in these glucosinolate fractions. So all Indian cultivars have almost about more than 100 micromoles of these glucosinolates, but these canola quality oil cakes should have less than 30 micromoles. So this is the standard levels of international levels which is acceptable for any oilseed crop to be consumed as a good crop. However, as I said, these glucosinates are also very important defense arsenal of mustard family. So how can we achieve? So in the last 2 decades or so, we have been testing different ways to breed low glucosinate in glucosinolates in case of mustard family members, and we have used conventional breeding approach, genetic engineering, as well as very recently we have been using genetic engineering approach. And I will try to convince you how genome editing has helped us in a very big way compared to these previous approaches where we have done a lot of challenges. With Professor Pintel and Professor Pradhan, long back, I started this work. I worked with their group, and we worked out, can we do this? Can we breed low glucosinate trait which is present in one of the non-Indian cultivar to Indian cultivars which are high in glucosinate? Can we breed it? So you can see this is quite a big pedigree. You keep on crossing this crop, this particular variety to this Indian type, then generate a line which is low in glucosinate, kept on crossing this with Varuna, recurrent genome, and every generation, almost 6 to 7 generations, you keep on crossing. The major problem with this crop, this trait, is that it's quite complex trait. It's quantitative trait. It's been governed by almost about 6 to 7 loci which we know at that time. So there are 6-7 major loci which control this complex trait. As a result, you need to breed all these 6-7 loci from this variety into this variety, which was a big task for us. Almost about 6 to 7 generations it took. But what we found, even after 6 to 7 generations, there was a big problem of linkage drag. So although you do a lot of conventional breeding through traditional breeding, we found there is a problem of linkage drag, that even after many 6 to 7 generations, only 75% of this Varuna genome we were able to have. Still there are 20-25% of this Hira genome was coming into the advanced generation lines. So this was a big challenge for us, that how to remove this kind of negative linkage of seed glucosinolates and the seed quality traits and the yield traits which is coming because of the linkage drag. So we thought, okay, let's try something using GM approach. We know that there is a transcription regulator. This is a pathway of glucosinate biosynthesis starting from methionine. You found that there are n number of genes. Luckily, we found there was a MYC28 transcription factor homolog in case of Brassica juncea, which regulate these pathway genes together. So this is a positive transcription factor which regulates so many genes. We did RNA-based silencing of this MYC28 sequence, and what we found that yes, in a single generation, we can generate lines which are less than 30 micromoles. No linkage, no problem of linkage drag, nothing. And what we found, we took these lines for event selection trial under open field condition. We found that yes, these lines are behaving very nice in case of for all the seed quality traits, only except the glucosinate trait which was coming down. So no compromise on seed yield and seed quality parameter, but the amount of time it took us to do even these event selection trial under open field condition was too much. Almost 2 to 3 years it took us to get these event selection trial permission, and now we will be taking these lines for BR level trials. But the kind of work it took us to take these to the field level condition, still we know that GM still have some lot of it's under regulatory issues. So, but in both of these approach, as I said, these canola quality lines which are reduced in glucosinolates in seed and in leaf also, they are expected to have, they are highly vulnerable to pest and pathogen, because what we are doing that we are reducing this defense compound throughout the plant. So what could be a better strategy? We wanted to have a solution which to develop an ideal line which will have only low amount of glucosinate in seed and retaining high amount of glucosinate in leaves, because so that these crops are not vulnerable to pest and pathogen, especially under Indian subcontinents. So we screened a big population of Indian mustard. We found that in all the population, there is no cultivar which is having low amount of seed glucosinolates and having high amount of leaf glucosinolates. Everywhere you find most of these cultivars are having very, very high amount of seed glucosinolates. We mapped this trait using genome-wide association and transcriptome-wide association studies, and we found there are many candidate genes which are promising base for this pathway. And luckily, we found one transporter gene which is known to transport glucosinate from leaf to seed. What does this do? In glucosinate, what is known that glucosinates are synthesized in leaves and then they are transported to seeds. We thought, okay, let's try this mechanism of doing the transport engineering. How can we use transport engineering to breed a mustard line which can accumulate low amount of glucosinate only in seed, but not without compromising glucosinate within the leaf areas or any other tissue types. But what we found is Brassica juncea is a crop which is allotetraploid from two different genomes, A genome and the B genome, and there are 6 homologs of GTR1 and 6 homologs of GTR2. So quite a complex engineering we need to achieve for this crop. There are 12 glucosinate transporter homologs in this crop which need to be tackled. But before that, we found that most of these transporters in fact are expressed very well, but catalytically we have no idea whether they are active or not. And there are no assays for transport activity to check also how we can do it. So one of my PhD students started working on transport assays to work out how to do it. Luckily, we found a cotton cell line. Cotton is a plant where these transporters are not expressed because these secondary metabolites are not expressed. So we developed cotton-based cell lines and we express these transporters there. And what we found when we express these transporters, we found that all these transporters, all the 12 transporters which we observed, were catalytically very, very active. They can transport glucosinate, different kind of glucosinate to a very high affinity. So these transporters are catalytically very active and they are also expressed ubiquitously. So this became our task even more challenging. Two of my graduate student, PhD student again work on this, and we worked through editing approach. How can we now, our goal is to edit all the 12 transporters, all to do mutation in all the 12 transporters. Luckily we found a conserved genome, exonic region especially in the second exon of the gene, which seems to be very much conserved, and we designed 3 guide RNA using multiplex genome editing approach. We transformed Brassica juncea and tried to in the mega variety of Indian mustard, and we developed lines which are editing. We found that these lines could do, this multiplexing can do editing of most of these transporters. Almost up to 10 transporters are edited in the advanced generation. There is a biallelic mutations for same kind of mutation in advanced generation. So this is something significant because most of the glucosinate in crop breeding, most of the genes which has been edited so far are only few, and this is the first success stories where up to 10 genes in a single plant has been edited by CRISPR-Cas9 based genome editing. And what was the effect of this transport? We found that the low amount in the seeds, the amount of glucosinates are now very, very low, less than 30 micromole of international standards in all the genome editing plants which we have generated. The leaf glucosinolates was not compromised at all. In fact, it was much higher because the glucosinate transport was no longer going to the seeds. So the leaf levels of these glucosinolates, these defense compounds are quite high in leaves. Also, very interesting fact was that for the first time we reported that these glucosinolates are transported from pod wall to the seeds, and pod wall also accumulate a good amount of these glucosinolates even in the edited lines. So this is something that, okay, it's a win-win situation that only the developing seeds are accumulating very low amount of glucosinolates, but throughout the plant, all the tissue types of this plant, we have high amount of glucosinolate, which is in fact very good for Indian subcontinent where you have a lot of pest and pathogen problem. So the benefit of these transport assays is that the engineering is that we have developed ideal canola quality mustard with improved oil quality. You have low amount of glucosinolates good for oil quality as well as for oil cake quality, improved seed meal quality for livestock and poultry feed, and also because of high amount of glucosinolates, these are more protected against pest and pathogen under field condition. And I will show you one data. We have through RCGM based guidelines, we have kind of taken these lines under open field conditions, done station trial, as well as these lines have also undergone ICR advanced varietal trial. And I'll show you just one data to convince you that on the left hand side you can see, on your right hand side you can see this, there is a conventionally bred canola quality lines which is heavily infected by termite attack during the maturity, but all our genome editing lines which are low in seed and high amount of leaf glucosinolates, they are intact, they are growing well, and they are forming very good seed set under open field condition in multi-location trials also. So this is something very significant that by engineering glucosinate, tissue specific engineering of glucosinate, we can develop edited lines which are very useful for for nutrition as well as for defense traits. Very recently, what we have done, not only we have reduced glucosinate from the seeds, another very important point, as I mentioned, that glucoraphanin is one of the nutritional glucosinate which is present in broccoli, and broccoli is consumed because of this particular compound, natural compound, which is known to be anti-carcinogenic. So we thought, okay, glucoraphanin, although glucoraphanin is very high in broccoli, but broccoli is something not very not very well cultivated in Indian subcontinent. But this Indian mustard, which is also used as a saag and oil cake, oil and oil stuff, can we try to engineer this particular glucosinolates in Indian oilseed mustard? We know there is one candidate genes which is AOP2 genes, which convert this glucoraphanin into these anti-nutritional compound, glucoraphanin and progrotin, which are anti-nutritional. So by editing, what we have done, there are 5 homologs of AOP2 in Junsia, and what we have done in that by editing 5 of these homologs, we have generated lines which are having very high amount of these green colored, if you can see, glucoraphanin, which are very, very high. Almost about 75 micromoles of glucoraphanin is now present in Indian oilseed mustard, which is the highest reported to date in any of the Brassica crops. So the benefit is that now this Brassica Junsia can be used as new generation of superfruit, which can have chemopreventive benefits, and also it will have lot of interstitial production of glucoraphanin and its hydrolysis product, which is sulforaphane. Regarding one last slide to show that yes, these glucosinolates can also be engineered for the defense properties. We know that in aliphatic glucosinolates there are different classes of glucosinolates, one which contain 3 carbon chain and another which contain 4 carbon chains. So these are 2 predominant class of glucosinolates in case of Brassica Junsia. My student found that, Bhanu found that when the C3 glucosinolates, particularly if you do some plate-based assays, these glucosinolates can take care of lot of pathogen effects. The growth of the pathogen Sclerotinia specifically is reduced to a very significant level, even at 1 micromole. But unfortunately, all Indian cultivars contain very high amount of these longer chain glucosinolate 4C. So can we try to engineer this? And we have done some very interesting work in 2019, where we've shown that there is one class of genes, protein methyl alkyl malate synthase. It converts short chain glucosinolates into long chain glucosinolates. So right now what we are trying to do is that there are 4 homologs of these methyl alkyl malate synthase gene. We are trying to achieve base editing of these residues which catalyze shorter chain and longer chain glucosinolate substrate so as to form only those glucosinates which can do only those MAM2 homologs kind of stuff, where we can have conversion only for smaller chain glucosinates. So that we can develop lines, Indian mustard lines, which can accumulate more of these C3 glucosinate in our Indian cultivars. So this is something which we are trying to work using genome editing for by precise base editing. We wanted to do this engineering for higher defense attributes also. To sum up, we have done some contributed something on glucosinate in biology in Indian oilseed mustard. In aliphatic glucosinolates, there are many areas which we have investigated. In fact, in all these areas, right now we are working on another compound, indole glucosinate or aromatic glucosinate. We have some interesting lead, and hopefully in next whenever I'll talk, I will try to express some of my expression of these stories which are coming in a very good way. Thank you very much, and I acknowledge here all my current and former director of BRIC and IPGR, lab members. I have presented work of few of my lab members, and many of them still there. My collaborators, Professor Deepak Mental, who has nominated me for this fellowship, Professor Pradhan, Professor Dr. Jagreet Kaur from University of Delhi South Campus, and many other collaborators, and funding from all different funding bodies. Thank you very much.
Thank you, Dr. Bisht. We have time for questions.
Good afternoon. I just want to ask, are there certain soil conditions that are needed to grow this crop, or are the soil present in the northern India, or any cultivable land is okay to grow this?
So this crop is well adapted to northern part of our country.
So the soil present is the soil okay, or does it need some specific conditions?
So this crop is quite very interesting because it doesn't require too much of I'll say nutrient stuff. It is a kind of, it can be used in low tillage condition also. But yes, sulfur is one of the compound which is depend heavily on that compound.
Another question I have another question. So since it's an empty phyloid genome, yes. So have you noticed any dominance in the subgenome in any one of the types?
Yes. So this is always true with us, because we have done a lot of subgenomic dominance studies in all our gene families. we found there are almost about 4 to 6 homologs. Especially GTR also, we found there are 4 homologs of GTR1 and 2 homologs of GTR, 4 homologs of GTR2 and 2 homologs of GTR1, which are always dominant. We have some data. I am not presenting it here, but yes, there is dominance. Yes. Yes, yes.
Another one, sorry.
Yes.
I just want to ask, like, when you, like, you know, is there like when there is a accumulation of GSL in like the vegetative tissues, have you noticed like cell toxicity, or like, you know, does it alter disease resistance or something?
So yes, disease resistance, I've shown you data on the disease resistance. They are very much tolerant against many pathogen and pest under open field condition. No problem on any of the yield parameters. These lines have already been tested under open field condition in our station trial. Also, ICR is doing independent trials in 16 different locations. And I'll just couple of days back, I got news that one of our lines has been going to the advanced varietal trial, third year also. Yes, thank you.
So I'm not from plant sciences, but when you do these field trials, do you also harvest the seed and see whether the taste is acceptable or the taste changes?
We do not go up to taste levels because right now the permission for consumption of these lines are not there. But yes, we do all the nutritional tests. We do glucoside analysis. Also, we do many different seed.
Okay, but the question is, will people accept it? Does it taste?
Oh yes, sure. Yes, because canola quality is still acceptable in India, and then we do expect that these quality lines will also be accepted. In fact, these lines are very good because globally there are no lines which are accumulating only low amount of seed glucoside and containing throughout all other plant parts. Yes.
So Naveen, very nice talk. Just was curious to know what type of mechanism GTRs follow, whether it's a uniport or a symport.
Oh, so this is a kind of this is an importer which transport glucosinate from the apoplast to within the cells, and we have done some these transporter assays to show that these importers can import these glucosinate substrate within the cells, and that was the beauty of those transport assays also.
And there is no effect of pH or
oh yes, they are pH dependent. They are generally, the requirement is about 5.8 is the optimal requirement, and we have done those assays also. In our plant method paper we have shown that. Yes.
Hello sir.
Yeah.
Sir, brassica is a crop involved in phytoremediation at times, like it is accumulant. So is that property affected by gene modification? Is it?
We have not tested up to those levels, but I'm sure these are very precise editing, and these transporters are doing job of only transporting glucosamides. That much we know about it. Yeah.
No sir, CRISPR also has some off-target
effects.
effects.
Yes. So this is very valid question. We have generated many lines, and I didn't see any problem with any of the other stuff. Right now, in fact, we are doing some deep sequencing of these lines to see if there is any off-target. And I'll get back to you.
Thank you, sir. Thank you.
All right. So if there are no more questions, let's invite the speaker.